Hello students, a warm welcome to all of you in ITLS Academy. In today's lecture, we will discuss about the protein structure database. Okay, the protein structure database is protein data bank. This is a PDB, that is a protein data bank. Okay, this PDB is used protein data bank. Okay, this protein data bank, okay, PDB. Why this protein data bank be easily retrieve the 3D structure of protein. 3D structure of protein. Okay, so we start today's lecture. We search PDB, then open this RCSB home page. Okay, this is the home page of PDB. Okay, here all information about the PDBs mentions. Okay, here this is a search bar. In this search bar we search protein structure. Okay, here I search human HBB protein, that is a human hemoglobin protein. Okay, and search here. These are the human HBB proteins. Here I search hemoglobin. Here I search a protein, that is hemoglobin protein related to humans. Okay, here loading. This is the result. This is the all query match of our search molecule. Here 22 lakhs 2938 structures are available related to the hemoglobin hemoglobin protein related to the humans. Okay, here we also refine or categorize the structure according to our requirement. Okay, these options are here available. We simply click and filter out and refine. Okay, here 70,807 proteins are related to human sapiens. Okay, here we also, if we want to experimental data, then simply search and refine. Okay, now these are the experimental result. Okay, I here I choose first one. Simply click here. This is the structure of hemomet meth hemoglobin, the variant of the theme. Okay, this is the name of this structure. This is the releasing date. This is the method by which we design this structure. Here the organisms. This is the macromolecules of this chain. This is the ligand. Okay, I simply click here. This is the PDB ID. Okay, first this is a summary page. Okay, this is the structure of meth hemoglobin, the variant of the theme. Okay, this is the database, or this is the structure summary. Here this is the this is the PDB DOI number. Here we also download the structure coordination in PDB, PDBX, PDML structure coordination in different formats. We also download these experiment, this X-ray data, X-ray diffraction data, and validation report and structure coordination. Okay, this is the structure summary. Sorry, this is the structure summary. This is the title of this structure. This is the DOI, PDB DOI number, DOI. This is the PDB DOI. This is the primary publication DOI. This is the entry author name. This is the initial deposition date. This is the initial releasing date, and this is the last revised date. Okay. Here, this is the all information about the experimental data. Okay, this is the method by which we design this, we extract this protein. This is the resolution, this is the R value, this is the value work, this is the observation. Okay, and start the starting modeling. This is experimental. Here we also view the detail. This is the detail. Okay, initial refinement, crystallization, crystal data, diffraction, data collection, refinement. All information related to the X-ray crystallography is mentioned here. Okay. Next is a literature or data related to this protein. Okay. Here is the entry ID. This is the molecule 1. Here A chain, C chain, E chain, G chain, and I chain. Number of sequence, organisms, detail of mutant genes. Here no genes, no mutations. Okay, this is the image. Okay, this is the sequence annotation. This is the sequence. This is the sequence from PDB, and this is the sequence from Unicode. This is the hydropathy. Hydropathy means which amino acids are hydrophobic and hydrophilic. Suppose this reason is, okay, this one. Number of amino acid is hydrophobic and hydrophilic. This is the disorder. Okay, and this is the PFAM or protein family. Okay. Next, this is the hemoglobin beta chain. This is a hemoglobin alpha chain. This is the detail about the hemoglobin alpha chain, and this is the detail about the hemoglobin beta chain. Okay, here also no mutation. This is the information about this beta chain. Here this is the PDB, this is the Uniprot, this is a hydropathy disorder bending, disorder binding, and PFAM or protein family. Okay, here is the information about the ligand. Here is the information about the experimental data. This is the length, software using, all information. This is the deposition data. Okay, this is the structure summary of our protein. Now we see the structure of this protein. Okay, this is the primary length of our protein. Basically, this is the amino acid sequences, and this is the protein. We easily rotate this protein and by the cursor. Okay, this. Okay, now this is the polymer chain, this is the ligand, this is the polymer and this is a ligand. Here no water molecule is present. Okay, this is the polymers, this highlighted one is polymer and this is a ligand. This portion is ligand, this one. Okay, now we also change the shape of our protein. Okay, here we click in three dots, then click in three dots, then add representation. Back one, wall, this is a Gaussian value. This is a backbone structure, this is a wall and stick. This is a cartoon representation of our protein molecule. Backbone representation. Will I delete here? Now I delete all. Firstly, I delete all these options. Okay. Okay. This is the protein. Okay. This is the cartoon orientation of our protein, our target molecule. Then this is the ball and stick. This is the Gaussian volume. This is the Gaussian surface. Okay, now if we want to see the ligands, then simply hide this. These are the ligands. Okay, this is the whole proteins or polymers. Okay, if we want to delete it, then simply delete. Now we want to see the ligand, then simply click on three dots and representation in Gaussian surface. These are the molecule. Here I remove. Okay, these are the ligands. Okay, these are the ligands. Okay, we easily change the representation of this molecule according to our requirement. Okay, next is the annotation. Domain here is the domain. This is the annotation information about this protein. Okay, here is the annotation, domain annotation by SCOP and SCOP classification, SCOP2 classification, ESCOD classification, CATH classification, protein family annotation, gene ontology, gene ontology, interpro on this is the Pharos or disease association. This is the domain classification according to the SCOP, structure classification of protein. Okay, this is the SCOP2 classification. This is the ECOD classification. Chain, family name, domain identification, architecture, possible homology, topology, family, and provenance version. Next, CATH, chain, domain, class, architecture, topology, homology. Okay. Next, family, protein family annotations. Okay, gene anatology information, glucose binding, small molecular binding, all information are available here. Here, protein family classification. Next, disease association. These are the disease associated with this protein. Okay, next is experimental data. Here, this all experimental data are mentioned. Okay, this is the all experimental data related to the our target molecule, and here we use the X-ray diffraction method. Next, this is the this is the sequence of our molecule or primary structure of protein. This is the A chain. This is the A chain classification according to the, this is the A chain according to the PDB. This is the A chain according to the UniProt. This is the secondary structure. This is the Ramachandran plot, and this is the rotamer outmer. This is the binding site for heme. This is the metal binding site. This is the, these all information are mentioned here. Okay, this is the all information related to sequences. Now this is the location of our gene in genome. Basically, the gene code for the hemoglobin is represent on chromosome 16. Okay, this is the information about the genome. This is the information about the gene. Basically, this gene is present on chromosome number 13. Okay, this position, and this is the position where this gene that code for the hemoglobin is present. Okay. Here the information about the gene where here the information about the gene where the hemoglobin coding gene is available on genome. Basically, this gene is present on chromosome number 13. Okay, and these are the version. Okay, okay, this is the version. Okay, we also, now we, this is the summary page. Okay, okay, here some error we expo here if we want to explore the structure, then we click here. If we want to sequence annotation, we also switch here. We, if we want to see the structure, we also click here and here also. If we want to know the sequence annotation, we simply click here and here. Okay, okay, here is the sequence annotation information. Okay, this reasons, this is the secondary structure of protein. Okay, this is the secondary structure of protein, this one. Okay, if we want to see the binding site of heme, then simply click here, this. Okay, if we want to see the binding site chain of B, then click here, here. Okay, if we want to see the hydropathy, then click here. These are the hydrophobic and hydrophilic amino acids. If we want to see the disorder, then simply click here. Okay, if we want to disorder binding, then I simply click here. Okay, PFAM. Okay, we easily visualize or annotate the our protein sequences. Okay, residues, polymer in track backbone. If we want to track the backbone, then we easily track the backbone of proteins. Okay, if we want to see the ligand non-standard, then simply click here. Okay, we easily visualize the all information related to the protein while they using these options. Okay, okay, we easily, if we want to see the amino acid alanine, then simply click here and we see the alanine. Okay, is an element symbol. Click here and see here. If you want to see the validation, and simply click here. The nucleoside basis, then click here. Okay, we easily see the all information about this protein according to our requirement. Okay, now move to back. This is the sequence annotation. This is the structure of our protein. Okay, this is the structure of our protein. Now if we want to see the electron density, then click here. Okay, this is the density of our molecule. This is the information about the density. If we want to see the validation report, then simply click here. This is the validation report. This is the alpha chain, this is the alpha chain, this is the beta chain. Here we also validate our results. Okay, this is the alpha chain, beta chain. In our protein clashes is also present. Okay, here the information about this classes is also available. Okay, now these are the ligands. Okay, this is the information about ligands. Okay, Jimmy. Okay, now this is the information about the polymeric chain. This portion is a, this blue one portion is a beta chain of our protein. This is a beta chain, and this is a beta chain. This portion is also beta chain. This portion also a beta chain. Okay, this portion is the alpha chain. This portion is the alpha chain. This is the hydrogen bond. This is the alpha chain. This is the alpha molecule of alpha chain. Okay, this is the photo. Here we also see the particular information about the protein along with amino acid and geometrical information, model, and also here we also see the all information of this structure protein. Why the hell? Okay, here we also here in this way we also validate our protein structure. Okay, this is the ligand interactions. If you want to see the ligand interactions, then this is the, here we simply remove this is the target molecules, and this is the surrounded molecule. These are the target molecules, and these are the target molecule. Okay, this is the target molecule, and this is a surrounded molecule. Okay, suppose this one. Okay, and these are the hydrogen bonds. Okay, if we want to see the, okay, okay, here is the geographical, no, sorry, global symmetry. Okay, if we want to see the global symmetry, then simply click here and we explore the global symmetry of our molecule. Okay, this is the unit cell, this, and this is the global symmetry of our molecule. Okay, now if we want to stereochemistry of our molecule, then this is the pseudo symmetry of our molecule. This is the pseudo stereometry of our molecule. If we want to find the similar assembles, then simply click here. These are the similar molecules related to our proteins, related to our target molecules. Okay, here is the information about the macro content. Okay, now if we want to display the file, then simply in FASTA sequence, then this we easily display this sequence in FASTA format. Okay, in PDB format. Okay, this is the PDB format of this protein. Okay, if we want to see the sequence in FASTA format, this is the FASTA format of this protein and this is the PDB. Okay, and this is the MMCIF format. Okay, if we want to download this protein, then simply click here, and these are the formats by which we download this file. So here I download this protein in PDB format. Okay, now our protein is downloaded in our PC. Okay, this is all about today's lecture. Okay, in today's lecture we see how we explore the protein structure in PDB. Okay, this is all about today's lecture. Please follow ITLS Academy on Instagram, Facebook, Twitter. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe ITLS Academy. Thank you.